Dear students, today we study about plant breeding. Plant breeding is the purposeful manipulation of plant species in order to create plant types that are better suited for cultivation and give better yields and are disease resistant. The list of the traits or the characters that the breeders have tried to incorporate into desired crop plants are like increased tolerance to environmental stresses, means they are capable to tolerate the harsh condition of environment like salinity, extreme temperature and drought. They must be resistant to pathogen and they increase tolerance to insect pest. They should be have high yield and improved quality of crop plants. Now we study the plant breeding program. Earlier methods of plant breeding involved crossing or hybridizing pure lines, followed by artificial selection to produce plants of desirable traits. The major steps in breeding a new genetic variety of crops are as: first of all, collection of variability. We collect the genetic variable plants. The genetic variability is the root of any breeding program. Pre-existing genetic variability is collected from the wild relatives of the cultivated crop, and these are evaluated for their characteristics and preserved for effective exploitation of the natural genes. The entire collection of diverse alleles for all germ in the given crop is called as germ plasm collection. Now the next step is evaluation and selection of parents. Evaluation of germplasm is carried out to identify plants with desirable combination of characters. The selected plant is multiplied and hybridized, and by cell pollination, pure line is created whenever desired. The next step is cross hybridization among the selected parents. The cross hybridization between the parents is done to produce hybrids that genetically create to give desired characters in one plant. Cross hybridization is a time consuming and tedious process because it involved the emasculation and bagging technique to transfer the desired pollen grains to the desired plants. The chances of the desired characters combining in the hybrid is also 1 in few thousands. Selection and testing of superior combinants. It involves the selection of plants among the progeny of the hybrids and desired combination of characters. The hybrid are superior to both of the parents, and thus this is called hybrid vigor or the heterosis. They are self-pollinated for several generations till they reach the state of uniformity or homozygosity. In order to avoid the segregation of characters in the future progeny. And finally, testing, release, and commercialization of new cultivars. The newly selected lines are evaluated for their yield and other agronomic traits of quality, disease resistance, etc. by growing them in the research fields. Their performance is recorded under ideal fertilizer application, irrigation, and other crop management practices. Now, after evaluation, the hybrid line is tested in farmer's field and the crop is grown at different locations in the country with different agroclimatic zone for at least 3 growing seasons. The tested material is evaluated in comparison to the best available local crop cultivar as a reference material for marketing of the material. And finally, the crop is released for commercialization, for cultivation. Thank you.